So today we are going to talk about the Human Genome Project. We are going to start with a quick video that it's like one of my favorite videos ever, and it kind of just explains what the genome is. So remember, the genome is all of your DNA, all of your chromosomes um, that you can see in a karyotype. That's your whole genome, all of your genes together. Okay, so I love that video. So it talks about um, basically everything that we have done so far in biology. Um, so that just tells you what a genome is. It tells you how important it is that we study the genome. Um, so that's what the Human Genome Project is all about. It's about studying the genome. Okay, so let's look at that a little bit more in depth. Sorry. Okay. So the Human Genome Project started in 1990, um, even before I was born. The Human Genome Project was started with the goal of determining the sequence of chemical base pairs that make up human DNA. So, scientists wanted to identify and map all the genes of the human genome. So, they wanted to map every possible combination of A, T, C, and G that could make up each individual person on this planet. Everyone has a unique combination of those four letters, which makes you you. And they mapped out what that looks like on a nitrogenous base level. So on the ATCG level, that's what they did. Um, if you look closely at this picture, that's what that is. It's A, T, C's, and G's. The Human Genome Project was established with the hope that knowing the human genome would allow them not only to identify genes that cause genetic conditions, diabetes, heart disease, cancer, etc., but also treat those diseases. I kind of explain it sometimes like if there is a person, if there's a like a murderer on the loose, you can't convict him. You can't put him in jail until you catch them. Okay, so the Human Genome Project is what catches these genetic conditions. It catches the criminal, but we can't quite treat diseases yet, or treat all diseases after we catch them, or else I would be cured of diabetes. But the Human Genome Project told us, okay, where did my diabetes come from? What gene was it on? What chromosome is it on? It identifies if you've got a lactose intolerance. What gene is supposed to make lactase? It identifies the gene number, the chromosome number, the exact place where that mess up in your DNA is. It found it. That's what the Human Genome Project is. So the Human Genome Project was successfully completed in April of 2003. And I had already been diagnosed with diabetes. I was diagnosed with diabetes in April of 2001. So when I was diagnosed, it was very frustrating because they had no answers. They had no idea how I had diabetes because it was a silent gene, and no one in my family had had it. Remember, it was activated by that strand of the cold that I got from somebody. I don't know. I didn't go to my class reunion, so I didn't ask anybody. That's why I'm still here and not in jail. But so that wasn't out yet when I was diagnosed with my disease, and so it was very frustrating not knowing where it came from, not knowing how to, I mean, even tackle the issue of is it going to be there for somebody else in the family? It was silent. So now I do know that it was silent and it was activated, like cell differentiation. It was activated by that strand of the cold. So, for the first time in 2003, scientists were able to read the genetic blueprint of a human being, of any human being. So, just as scientists hope, this project has been useful in determining carrying genes and developing gene therapy. So, now if we can identify these genes, you can get tested before you ever decide to have kids one day to see if you are a carrier of any genetic diseases that may change your mind. That's one of my reasonings as why I don't want kids, because I don't want them to have diabetes. I wouldn't wish the pain that I go through on my worst enemy. And so, knowing what I have and knowing that it is genetic, I don't want to pass it down. Um, so, gene therapy is a method of using genes to treat or prevent disease. Nucleic acids polymers inserted into cells as a drug are used to express proteins or interfere with expression. We're going to talk about an example of that in a minute. Remember, DNA codes for proteins that determines traits. Uses include immunodeficiency. So, an immunodeficiency disease is one in which disorders prevent the body from fighting infections and diseases the way that it should. So, one of those is cystic fibrosis. Another is called SCID. These are the types of diseases that, if you have one of them, then you should not be out in public right now because of the coronavirus. So, it's used in fighting infections. Your immune system, and if it's low, you can't fight those. So gene therapy has helped us with this, though, and I'm going to show you the way um, in this next short presentation. So this is just a little bit of extra different way of saying what the Human Genome Project is. A genome is an organism's complete set of DNA. It was a 13-year effort to sequence all base pairs that compose human DNA. So basically, they can look at a map of my A, T, C's, and G's that if they took my genome, they could recreate, or if they could actually recreate my combination of A, T, C, and G to say, okay, this would make up Lindsay Formwalt. So they can do that for you too. Um, to produce, the goal of the Human Genome Project was to produce a gene map that shows the relative location of each gene on every chromosome. Advances modern biotechnology processes like genetic engineering and gene therapy. And it also, another goal is because scientists and doctors have to know the exact location of particular genes. You've got to know the location of the bad guy before you can go get them and take them to jail, before you can treat them. So I have this picture in my room. Um, so it's hard to see, but this is a poster. You see, it's got 1, 2, and it's got through XY. So this is 23 chromosomes, and the little tiny words beside that are all the diseases that are on that one gene. Okay, so I wonder if I can actually zoom in and see if I could. Nope, I don't think so. Nope. um, but here is one thing that you can see on this. Okay, sort of. Actually, I've got it back on here. Okay. Okay, here is chromosome 21. So this is a close up of the 21 from this page right here. Um, so this has 46 million base pairs, and it's one of the smallest chromosomes. So that's 46 million combinations of the four letters A, T, C, G. That's a bunch of A, T's, C's, and G's. Okay, that's only on one chromosome. You have 23 of these in each one of your cells, and you have what was it, like trillion cells? That is a crap ton of A's, T's, C's, and G's going on in each one of your cells. Okay, like this is just mind blowing to me that they can actually figure out each possible combination when one of the smallest chromosomes has 46 million base pairs. That's why it took 13 years. So you can see um, on chromosome 21, you guys know that this is Down syndrome. If there's an extra chromosome, that's the Down syndrome gene. This is also the chromosome that carries epilepsy. Let's see if there's any other ones that you guys would know. Alzheimer's on chromosome 21. Leukemia, leukemia that comes from Down syndrome, is on chromosome 21. So, like, those are all the diseases that they traced back to chromosome 21. There's chromosome 21 is made up of 46 million combinations of A, T, C, and G. So I think like chromosome 1, which is one of the biggest, is like I was trying to look at it on here, but I couldn't find it. It's like 300 and something million base pairs. It's a lot. So that's, I mean, that just of that amount of those four letters makes up who you are. and they mapped out each different possibility, each combination that that could be. So that's what the Human Genome Project did. That's why it's so groundbreaking. That's why it has so many uses and it took so long, but it gives so much hope. You know, we may not be able to treat type 1 diabetes, but we know which chromosome it's on. I'm pretty sure the type that I have is on chromosome 15, if I can remember correctly from my math in class. And you know, it's just hope. It's just hope that they know where it's at, so maybe we can treat it. Here, in a second, I'm going to tell you about a couple of gene therapies that they like for you to know about in the standards. But this is really cool, the gene therapy that they do. So cystic fibrosis it's the production of abnormally thick mucus leading to the blockage of the respiratory and digestive systems. And you don't have this in your notes, so don't worry about it. Just listen. Okay, so here's what they've done with cystic fibrosis. They they made a they developed a technique that enables patients to breathe in molecules of DNA, delivering a normal copy of the defective gene to the cells in the lung. Okay, so it's like an inhaler. They inhaled the correct DNA, the corrected gene. They have cystic fibrosis gene. They inhaled the correct gene. That gene makes proteins, right? Gene to protein. Okay, and it helps the lungs function properly until the body fights off that defective gene. The body at some point says, hey, that doesn't belong to us. We're going to get rid of it. Okay, so with this gene therapy though, that's just inhaling something that takes the lifespan of someone with cystic fibrosis from dying in their 20s to living a normal, semi-normal life probably into their 40s. So I mean, that is amazing to me. So the gene makes a protein that controls the movement of salt and water in and out of your body's cells. With CF, the gene makes a protein that doesn't work well, causes thick, sticky mucus, and very salty sweat. So it wants to hold the salt in. And so with the corrected gene makes homeostasis happen in the lung cells. So that's just one example of what the Human Genome Project has done. That is how, if you guys remember me talking yesterday about bacterial transformation, that's how we know where the human gene to make insulin is, so that we can locate it to insert it into bacterial DNA. Another example of a immunodeficiency disease is called SCID, which is called severe combined immunodeficiency, a group of inherited disorders that causes severe abnormalities of the immune system. So this is X-linked, so it's sex-linked. It's on the X of the XX or the XY on the 23rd chromosome. Doctors can inject healthy copies of the gene into a patient's bone marrow stem cells. Okay, so those stem cells regenerate, and if they regenerate with the corrected gene, it can help the immune system to strengthen. Now, that immune system won't stay strengthened. That's why they have to keep getting injections to kind of keep it strengthened. So, and that's in bone marrow. So that one's still very painful, just like diabetes is painful, and it's treatable. But this is a way that they are isolating that gene so these people with SCID can live longer. All right. Um, I think, yeah, that's it. Tomorrow or next week, we'll talk about the ethical issues of genomics and biotechnology. Have a great day, guys.